Hello, thanks a lot for stopping by my poster. I'm Siddharth Uppal, and today I'll be telling a story about how we discovered a biosynthetic gene cluster and the bacteria harboring that gene cluster for the production of a potential anti-cancer drug. So let's get started. Our story begins back in 1990s where a group of researchers actually took a deep dive into the Gulf of Mexico and found out or extracted a sponge called Forsythia. And when they extracted all the natural compounds from this sponge, they actually found a molecule which they named Lasonolide A. And when they investigated this molecule against different cell lines, they found out that wow, this molecule has a really potent anti-cancer activity in nanomolar concentration, which was pretty awesome. And they were like, yeah, let's take this compound to clinical trials. We can make this an actual anti-cancer drug. But as soon as they decided to do that, they found themselves stuck in another challenge, which was, well, the supply of this compound is pretty limited. The reason is because the sponge Forsythia is the only natural producer of this compound, and as you can imagine, we can't just keep using the sponge to extract Lasonolide A. It's just not sustainable. Then they decided to call the organic chemist and ask them to synthesize this. Well, a number of organic chemists tried, but the process was super long and the yield were not super great. So they were stuck, as to say. And that's where we come in. We were like, we can actually use metagenomics or bioinformatics to actually identify the producer of this molecule or the genes that's making this. And once we have that, we can put the genes in another bacteria that is heterologously express it and then produce this molecule or try to cultivate the bacteria that's making this molecule. And that's where we come in. So the two research questions I decided to find out was, how is Lasonolide A being produced and what is producing Lasonolide A? So we did whole genome metagenomic shotgun sequencing of the entire sponge, got the reads back, assemble the reads into contigs, and then bind the contigs into metagenome assembled genome. And when I analyzed that data, I was actually able to identify the entire biosynthetic gene cluster for the synthesis of Lasonolide A, and that's shown here. We named it LASPGC. Each arrow here is a gene, and I was actually able to go through each gene one by one, annotate different domains, and identify the entire biosynthetic scheme for the synthesis of Lasonolide A, which is shown here in the middle panel. And this biosynthetic scheme turned out to be a trans-AT polyketide synthase pathway. Some of the cool stuff that we see with each trans-AT polyketide synthase pathway was also seen here. We had a monooxygenase domain that inserting an oxygen here, and this monooxygenase domain had the same modular architecture of KS, DH, KS, DH, monooxygenase, ACP, and KS that was recently found by Jan Peel. Plus, we had two pyran synthase domain. One is here, that's required for tetrahydropyran ring in the Lasonolide. Plus, another unique feature was the presence of this condensation domain as a termination domain and not a thioesterase domain, which is generally seen with PKS pathways. So that was pretty cool too. And once I had the whole biosynthetic scheme, I wanted to dig deeper into the bacteria that actually has this contig. And that takes us to the rightmost panel here. So the graph here is a dimension-reduced graph of a 512 Kmer frequency table with coverage as a z-axis, and each dot on this graph is a contig that we got by assembling the reads. The contig in the red is the Lasonolide gene cluster that's shown in the middle panel, and all the contigs or the dots on the blue are the genome that has this gene cluster. So one of the first thing that we see from this graph is that the red dot is super separate from the blue dots, or the LASPGC has a really different nucleotide composition from all the contigs in the genome. And this led us to hypothesize that maybe LASPGC is being horizontally transferred. And the reason we think it's being horizontally transferred and not incorrectly binned by us, because we verified that LASPGC actually belongs to the genome using PCR, mapping of assembly reads, and assembly graph as well. Another thing that we see from this graph is that the coverage of LASPGC, which is the z-axis, is pretty higher than the average coverage of all the contigs in its host genome. To say mathematically, or when we analyze the actual coverage, the coverage of last BGC was thrice the coverage of its host genome, which led us to suggest that maybe last BGC is actually repeated thrice. And we actually validated the hypothesis using PCR, assembly graph, and mapping of paired reads, and we actually found 3 connections and verified that's, yeah, it's actually repeated thrice, which is pretty cool. Finally, I decided to analyze this whole genome that has Lasonolide gene cluster. And when I analyzed this, I found that this is actually a novel genus under the phylum Verrucomicrobiota. And when I found, wow, this is a new bacterium, I want to name this. And I did that. I named it Candidatus thermopylae lasonolidus. Candidatus because it stayed uncultured. I found this by shotgun sequencing. Thermopylae is actually a tribute to 300 Spartans who fought at the Battle of Thermopylae. So there were 300 Spartans, and our BGC is repeated thrice. The Spartans fought to protect Persians from their kingdom, and we hypothesized that the bacteria is producing Lasonolide A to protect the sponge from any predators. So that's the analogy. And Lasonolidus because producing Lasonolide A. So that's the story, my friends. I'm Siddharth Uppal. My PI is Professor Jason Kwan at the University of Wisconsin-Madison. This project was completed in collaboration with a number of people listed here, all from Florida Atlantic University. And feel free to contact me. I love talking about bioinformatics. Any potential collaborations, feel free to contact me. And once again, thanks a lot for stopping by at my poster.